I just want to say thanks to everyone for coming, whether you've been here before, this is your first time. It's really fantastic for us to have so many people from the community interested in this lecture series, so we're thrilled to have you all here. Can I just, for grins, get a show of hands of who this is a first time here at UCI for? So a lot of people have been here before. That's great, because I changed up my intro talk, so you guys will all be pleased to know that. So just as a broad overview, in case you don't know, this is the Sue and Bill Gross Stem Cell Research Center. We're over 50 faculty that spans 6 schools, 21 departments on campus, so we're a very interdisciplinary center. That's our focus, to really be a bridge across the university as a whole. Our missions are discovery, basic science, on up to clinical translation, to teach, educate high school students, undergraduates, graduate students, medical students, residents, in fact, here, and to heal, to really be a source to drive clinical trials forward. And this is largely representative overall of the mission at UCI and certainly within the College of Health Sciences. So, a little bit of more of a bold statement than I often make here. I think stem cells, the people who work in this building believe, really will revolutionize the treatment of human disease and injury. And in fact, they have already. And so I've tweaked this presentation a little bit to try and give you guys a little bit more of a sense of how that's the case. So this is a cartoon you may have seen from Dr. Golub before, kind of prefacing where science with stem cells might go. Fill her up, mate, right? That the possibility that stem cell replacement and stem cells of treatments is really coming to fruition now. This is a review that appeared last summer in the New Yorker, which if anyone here is interested in a more general discussion of stem cells and where treatments are, what their potential is for the future, is a great source. What I really want to use it is just as a slide to highlight the idea that there's incredible promise here, but there is the potential for a price. This is about the dollar value sign price of stem cell therapeutics. But I want to use it to highlight the distinctions that I think are really important to make between what happens in the clinical enterprise that we have here. We have Dr. Daniela Bota, who's our clinical director for our Alpha Stem Cell Clinic, speaking tonight, and the structure, the responsible clinical trials that happen through that network, versus what you have heard about from Dr. Cummings before and other folks in our lecture series, the much less regulated, if at all regulated, markets that are around us in the surrounding area. And so stem cell clinics that really can be problematic in terms of making claims for therapies that aren't quite there. So just a highlight on my part. And in that regard, I would point you to this lecture, which is part of our video files for the center. That was our kickoff lecture for this year, Stem Cells, Hope and Hype. It's a great resource. Also a fantastic resource is a website called A Closer Look at StemCells.org, just one word. And that's a website that's run by the International Society for Stem Cell Research. It's a really great resource in terms of thinking about distinguishing what is an actual clinical trial from unregulated hype, for lack of a better word. Again, the last thing I want to point you to here is just the video series that we have that have come largely from these lectures. So if you're interested in any of the other topics, I would point you to our website because I think we are developing a nice set of resources there. So part of the question that I want to highlight then is, how do we separate hope from hype in the context of stem cells? And here again, highlighting the wild variety of claims that are made by stem cell clinics in the area and in fact across the country within the US now for things that are claimed to be treatments in the absence of really empirical scientific evidence to support. So one way that I think is important to think about that is that this road to regenerative medicine and stem cell treatments really in California has started with the California Institute for Regenerative Medicine, leading on into the clinical initiatives and the alpha stem cell clinics that are a part of that network and of which UCI is a part. You'll hear a little bit more tonight. So we are at the Stem Cell Center a CIRM Institute. What is CIRM? It's the California Institute for Regenerative Medicine. Many of you, I think, must know at this point that this was brought into being in 2004 when Californians decided as a group to put into place a ballot initiative, Prop 71, that provided $3 billion in funding over roughly the last 13 or 14 years for stem cell research. Again, there are a lot of policy issues that led to this. We have a video on that was presented by Dr. Golub at the end of last year that really outlines the background for how that came to be. But the important part of what CIRM does is that its goal is to fund stem cell research at institutions and, in fact, companies across the state with the goal specifically of accelerating treatments to patients where there is unmet medical need. And so, for example, at UCI, kind of our particular expertise, this falls into the domain of neurological disorders because there's such a great unmet medical need in terms of what conventional pharmaceutical treatments have been able to provide. So in terms of where we are now for status in that regard from CIRM, there's actually over 60 CIRM-funded clinical trials across the state, more than 70 clinical trials easily that are being run through the Alpha Stem Cell Clinic Network. What is the Alpha Stem Cell Clinic Network? It's 6 centers that are distributed across Northern and Southern California, one of which is UCI, through which these 60 clinical trials are largely being organized, and you can get a sense of what the distribution of diseases are here. Everything from ALS to retinal disorders to diabetes. In fact, at UCI, just 2 weeks ago yesterday, we did our first transplant for a clinical trial using stem cells as a testing for a treatment for diabetes, right? So a wide array of what those trials look like, and we're very excited to be involved in that. This is our current Alpha Stem Cell Clinic pipeline that's run by Dr. Groisman and Dr. Bota, who are here tonight. And we're very excited to be participating, not just in the basic science side, but the translational pipeline for these trials. There we go. So what has CIRM achieved in that regard? One of the things that's been incredibly exciting is actually things that many of us who are stem cell researchers probably never thought had the chance to be the case within the period of funding that CIRM originally intended, just 10 short years. So this is an example. This is Angelina Vaccaro, who has a genetic disorder called severe combined immunodeficiency disorder. So her immune system fails to develop normally. If you have ever seen the John Travolta movie in the 70s, Boy in the Bubble, this is that disease. And this is an example of an incredible win-win for regenerative medicine and stem cell treatments, because it's possible to target the hematopoietic stem cells where this genetic defect resides, replace the genetic defect, and actually result in a cure. So at this point, this man, Don Cohn from UCLA, again through the Alpha Stem Cell Clinic Network, has treated over 40 kids with this disease, many of whom are living now a completely functionally normal life. And that's an amazing thing to have achieved with regenerative medicine through the efforts that are being conducted in California. More locally here, focusing on UCI, just 2 quick examples. We have right now a clinical trial kicking off with Dr. Henry Claassen for retinitis pigmentosa. So this is a clinical trial that's testing stem cell-based therapy that was developed entirely in-house here, now moving through towards patients, hopefully eventually being applied also to macular degeneration. And you're going to hear tonight from Dr. Bota about her work with glioblastoma and trying to target cancer stem cells as one way, a different aspect of regenerative medicine, but still a stem cell-based therapeutic, a stem cell targeting therapeutic. So we're very excited about this work through our Alpha Stem Cell Clinic. More largely speaking, broadly speaking, in terms of that original ballot initiative and what's been accomplished by CIRM, it's pricey, $3 billion. But in an economic report that was issued through the University of Southern California last fall, the estimates are that almost $11 billion of additional gross output to California was added to our economy through that $3 billion investment. And so I think that's important to realize that this type of funding can really be a driver also for our economy, in addition to the benefits that may be gained from a translational and clinical perspective. So hype is easy in that sense. This is what we see at the clinics that I'm referring to within the area that are claiming treatments without really having that evidence-based medicine. But how do we get the hope part of that started in our hope versus hype comparison? And here is some information that many of you will have seen before, but I just want to emphasize because I think it really bears thinking about in the context of how we move forward in 2020. Like most of what runs in the world, hope always starts with a pipeline. In our case, that's a translational pipeline when it comes to research. So we have to have a discovery basis where we're investigating the very basic science of discovering stem cells and how they work. moving on through the very early stages of translation, things that we do at the bench, and into actual clinical trials. And again, that's what the Alpha Stem Cell Clinic really is all about, both for CIRM and for UCI. And in that regard, it's important to note that research is traditionally supported by the federal government. And so many of you have seen me show this slide before, but I can't emphasize enough how much funding for research has declined in the United States since the early 70s when, for example, we started the war on cancer. We're at 1/2 of our funding rate nationally right now. So we need to have other ways to be able to drive that translational pipeline forward. It's absolutely critical. The last point I want to make in that regard is that the funding of universities like UCI is critical both to generate new companies. This is an example from a FASEB website, all new companies that were generated out of the idea of that translational pipeline, and for the generation of new treatments. So again, this is an example of new drugs that have come to market just within a 5-year period for this particular study, showing that federal funding or public funding of some kind contributed to each and every one of them. And so that funding pipeline, that basic science to translational science to clinical science pipeline is really critical. At the Stem Cell Research Center, we try and help that happen through a seed grant program. This was started under Dr. Sid Golub, the previous director here. And the goal is, with that declining set of sources of federal funding broadly, we want to make our investigators more competitive to be able to get that funding and drive their programs forward. So the way that works is we've provided in this 5-year period about $450,000 of seed funding, and that's helped our investigators to bring in over $11 million of federal and state funding. So that's a 25-fold return on investment for us. So this is a program that's really near and dear to our hearts, and which I hope you all will think about. So what has that led to? So this is Matt Lurton-Jones, for example. He's a CIRM-funded researcher. He also had a seed grant to be able to develop these projects. He works on Alzheimer's disease and developing induced pluripotent cells, human-induced pluripotent cells, to study Alzheimer's disease, working back and forth between human systems and mouse models. Leslie Thompson is another one of our investigators who has been able to garner a large CIRM grant actually at the clinical stages of development of a potential stem cell treatment for Huntington's disease, preparatory to moving forward into clinical trial. And Brian Cummings, who's here tonight, who has a translational award from CIRM for that same purpose, focusing on traumatic brain injury, with a seed award that came through the Stem Cell Center all the way through the basic science pipeline into the translational development of that therapy. So those are projects we're all particularly excited about. So how can you help? Well, it can be anything from do your homework. So I would encourage you to go to the Americans for Cures research website and educate yourselves about CIRM and the upcoming ballot initiative for 2020. If you support stem cell research, please consider contacting your state and federal representatives to indicate that support, especially at the national level. We're actually under threat right now in terms of much of the stem cell research that we do in California, and there are some limits being placed on what that funding can be. So this is very important to us. Please sign up for our newsletter or follow us. We are looking for volunteers all the time to help us host these sorts of events and to do additional public outreach into the community. Please consider making a gift. And I would just note that research drives the economy. I hope I've given you an example of that in terms of how CIRM funding has benefited within the state. But actually, less than 4% of philanthropic donations across the country are estimated to go towards research. Less than 4%. So this is something that can help your community and can help this translational pipeline. Last, I just want to end on focusing on what our upcoming schedule is. We have a few lectures left yet this year. So we're here, radiation biology and stem cells tonight. Next is March 24th, seizures in a petri dish, looking at modeling and insights into epilepsy. And we're excited to have that be our next lecture in the series. So thank you all for your attention. Thank you for coming tonight. And with that, I'd like to introduce our two main speakers. Munjal, are you going first? So Munjal Acharya is an in-residence faculty here who's a tremendous researcher. He's been wildly successful at UCI. Come on up, you're going to have to help me change your slides. We've been pleased to have him participate in the center and be a part of the center. He's been incredibly successful as a young investigator in terms of garnering independent funding with a really interesting line of work in terms of understanding how stem cells and stem cell products might be able to treat the consequences of our cancer therapeutics. So with that, Munjal.
Thanks. Thanks, Aline, for the introduction, and thank you for all for spending time tonight. I hope it will be much interesting than democratic debate. So tonight I will talk more about radiation effects or radiation therapy effects on brain function. How can we use stem cells as a therapeutic agents to reverse those brain injury induced by cancer therapies? And what are the recent advancements in more technological development to treat brain cancer such that it does not induce normal tissue toxicity? So at UCI, as Eileen mentioned, we are more than 50 researchers associated with Stem Cell Research Center. We are from Department of Radiation Oncology, and from past decade, we have begun research on how radiation affects CNS or central nervous system or brain function. From past 10 years, we have been doing research on mechanisms that lead to these brain injuries in order to find out cure to reverse this disease condition. We are approaching by various ways. Of course, using stem cells or stem cell-derived nanoparticles or nanovesicles called exosomes. We also use drugs to reverse those conditions. We apply certain genetic approaches to study the mechanism. And we have recently developed ultra-high dose rate flash radiation therapy, which is just begun at the UCI with the recent National Institute of Health funding. So this will be the three main part of my talk. The first part would be to discuss what are the basic science level, how radiation affects brain, why radiation is important for the cancer therapy, and how it affects brain structure and function. If this radiation therapy affects brain function by damaging certain pathways or cellular machineries in brain, can we reverse that or regenerate the disease situation or injured brain using stem cell therapy? And what is the buzz about the most recent radiation therapy, which will be kickstarted in the US soon. So this is a general slide provided by National Cancer Institute. What they say, on x-axis is the percentage survival of patients after cancer therapies, when they receive cancer therapies. Compared to 1970s, if you talk about current situation, the black bars, what it shows, the cancer number of patients surviving more than 20 years after cancer has been increasing. So people don't die immediately after cancer because of recent advancement in oncology research, chemotherapy research, or latest advancements in immunotherapy research. People survive more or long after cancer therapy, and that's why cancer has become a chronic disease. So we have more cancer survivors in US compared to the past 3 decades. For example, in case of breast cancer survivors, we have more than 3.5 million breast cancer survivors in the US who may be suffering from the condition called chemo brain or chemotherapy-related cognitive dysfunction, which affects their day-to-day life. Similarly, for children who are affected by cancer in their early childhood, who are given radiation therapy, has been increased a tremendous number, and those children who are suffering from chemotherapy or radiation therapy-related cognitive impairments or brain injury has been increasing from past 2 decades. That's why cognitive dysfunction or behavior or learning and memory function after radiation therapy or cancer therapy or chemotherapy is particularly important for the quality of your life for the cancer survivors. So with exception of survival, cognitive function is one of the most critical criteria for developing therapeutic strategies to kill the cancer. So you want to definitely kill the cancer and make that patient to survive long, but you also want to improve the quality of life of those children, those women, those patients who will be able to conduct their day-to-day routine. So first question we ask, why radiation therapy-induced cognitive dysfunction is important to study? So either brain tumor, what we call glioblastoma or glioblastoma multiforme, adult or pediatric cancer patient, they develop progressive dementia. Dementia means forgetfulness, loss of memory, loss of cognitive function. And certain factors are involved that may perpetuate into this situation. Now at UCI, we use this is most commonly used radiation paradigm throughout the US. This machine is called LINIAC or variant LINIAC called rapid arc system, where from the head of this machine, radiation source delivers radiation to the tissue. So it actually delivers certain radiation source to the brain volume to kill the cancer. There's a lot involved in this treatment planning. There are physicists involved, radiation oncologists are involved, neuro-oncologists like Dr. Daniela Bota who are involved in treatment planning before we actually come to this situation in order to deliver this radiation therapy to the patient. So in order to study this similar situation called clinical scenario, in our laboratory we have developed rodent model of radiation therapy-induced brain injury. So we did same thing. We used the same machine. This is rodent brain or rat or mouse brain. And using the treatment planning plan like MRI or CT scan, we can generate this dose volume painting. So what you see here is called dose volume painting. And the red circle is where you will deposit maximum radiation dose to kill the cancer. It also generates epicenter around the dose volume, which is actually normal tissue. For example, let's say you have cancer with the periphery of only 5 millimeter of diameter. However, when you radiate that part of the brain, it also spill out to the other parts of the brain, which is important for normal function, which is important for your daily learning and memory activity, which is important for your working memory or memory recall or forgetfulness sort of function. So we have developed this model to study the effect of radiation on brain function, and that can be further expanded to bilateral. Bilateral means both sides of brain can also be irradiated, and it can also be studied. So what we found so far, this is just a summary from past 10 years of research shown here. I'll explain some of the research in my slides. When you expose brain to radiation or when you treat the mouse or rats with chemotherapy, which has been used in cancer therapies, it leads to progressive decline or reductions or injury into fine structures of neurons. So what are neurons? Neurons are the functional units or cells in the brain that connects with each other to form a neuronal circuit. And connectivity between neurons in the brain is responsible for learning, memory, storage, and recall of your memory. So how smart you are, it depends on how this circuit is formed. Each neuron forms intricate circuitry in brain, which are interconnected with each other. And when you irradiate or when you destroy this circuitry, it leads to dysfunction of brain. Each neuron also forms memory units called spines or synapses. These are the small structures on the neuron where synaptic input or whatever you see, observe, or learn is stored long term after you learn particular thing. So such fine intricate structure of neurons are affected by radiation or chemotherapy. That leads to decrease in a dendritic complex. Dendrites means this long structure here you see is dendrite. Spine means memory units, what you see here are spine, and their morphology, how they store the memory. It has been led by, stored by synaptic proteins, which are lost after radiation or cancer therapies. And we also see major increase in inflammation. Now, inflammation is key here, which will also be explained by Dr. Bota. When you irradiate or expose brain or system to the radiation or chemotherapy, it produces reactive oxygen species. These reactive oxygen species are formed from water. They are damaging oxidative stress. For example, many of us take antioxidants, right, in order to decrease inflammation in our body. And same reactive oxygen species can cause inflammation in the brain. And the brain is called Neuro Inflammation. It triggers activation of brain immune cells called microglia. We know WBC is in blood, right? Similarly, in brain, we have microglia. This microglia become inflamed. They attack this fine neuronal structure that leads to the damage. In order to study such damage, we use a transgenic mouse, green mouse called Taiwan EGFP mouse. GFP means green fluorescence protein. This protein has been isolated from jellyfish, which is it actually fluorescent protein after exposure to UV light, and it produces green color, and you can actually see that under confocal microscope. What you see here is a memory center of mouse called hippocampus, and hippocampus has a functional circuitry of neurons. What you see here, round structures are cells here. And what you see here are dendritic structure, which is emanating from the hippocampus. And it interconnects various neurons in the brain to form learning and memory aspects. So when we take this mouse and expose them using our same machine that we use for the patient therapy, and you just irradiate hippocampus, what would happen in brain? So let's see that. When we put this same neurons or slices of this brain in confocal microscope or laser scanning microscope, you can actually construct a structure of neuron. So the blue balls here are neurons. The green dendrites are emanating from neurons that forms complex dendritic tree, and their red dots are spines. So when you irradiate brain, you can actually easily compare. Look at the left, which is a left-hand bar. This mouse is not exposed to radiation. This mouse is exposed to very low dose of radiation. And the last slide here, this mouse is exposed to the radiation dose, which is equivalent to use in the clinic. And you can actually clearly see how dendritic structure degrades after exposure to radiation, right? So this is fine dendritic structure which has been degraded after radiation exposure. When we focus further to define the dendritic spine, remember dendritic spine are the structure that stores the memory or synaptic function. The spines can be defined into four or five different types. Long or philopodia spine, which are immature spine, which actually forms when you learn something. Mushroom or stubby spine are mature spine, which is important for memory recall processes. And in each case, we see loss in the formation of new spines after radiation exposure. This can be seen very detailed here. In this case, mouse is not exposed to radiation. This is 10 days after radiation. This is 30 days after radiation. You can clearly see the synaptic loss or spine loss in terms of loss of red spots, which are memory units in the brain, and also loss of dendritic structure, which are green parts in the brain. So radiation not only affects the whole neuron structure, but it also reduces the red spots or dendritic spines that forms memories. As I said before, radiation or chemotherapy exposure induces reactive oxygen species, which actually triggers gliosis or inflammation in the brain. In the brain, like WBC in blood, in brain we have microglia that forms immune function in brain. They are activated. And this activated microglia or hypertrophic microglia or astrocyte, which are important for metabolic function, are triggered called hypertrophy or gliosis that actually attacks these neurons and reduces the number of spines in the brain. So far what I have shown you that after exposure to radiation or chemotherapy, it leads to huge damage to neurons, spines, and memory units in brain, and which is associated with inflammatory signatures. So what can we actually do to address this unmet medical need? We asked this question about 10 years back when we were given funds by CIRM, as Eileen mentioned. So based on our first CIRM grant, we started this research using stem cell-based therapy, because what we hypothesized that radiation exposure could lead to loss of neurons or structures in the brain. If we simply transplant stem cell, can we replace those lost cells? And if we replace those cells, can we revert the cognitive loss or behavioral loss? This is a summary of our 10 years of research, which is summarized here. We can use multiple types of stem cells to be transplanted in radiated brain that can actually reverse cognitive dysfunction caused by radiation. It is long-lasting, and it can affect multiple learning and memory centers and network activity in the brain. We measure this function using neuron structure, synaptic loss. Stem cell grafting also reduces inflammation in the brain very significantly, and it also protected neuronal morphology. When we dig further into detail, it was actually not cell replacement, it was more of a trophic support. Whatever stem cells secreted in the brain after transplantation might have reserved the cognition. To summarize this, let's say these are stem cells. See the middle panel here, red cells or red structures. They are transplanted stem cells in mouse brain or rat brain, which is radiated. After transplantation in brain, stem cells can decide remaining stem cells, or they can also differentiate into neuron. Differentiation means stem cell can also form neuron or glia in the brain by functionally integrating into damaged brain. So if you see very small number of stem cells are actually form neurons, which are shown here, about only 5 to 10%, and majority of them form glia. And when we counted each stem cell after transplantation in brain, which takes about 6 months to generate this graph, immediately 1 month after transplantation, only 25% were surviving transplanted stem cells. And by 8 months, we only have 4% of transplanted stem cells surviving in the brain. So we do not actually see cell replacement. It's not actually cell replacement. However, when we check the transplanted stem cell, what they are doing, they are actually forming a functional circuit in the brain. So this red structure, what we see, is called behavioral plasticity gene called ARC. ARC is expressed when you put mouse from cage to let it explore to new toys or objects. So those neurons fire. And when we check our transplanted stem cell, which is here, it was also integrated in firing. It was also making neurons. So only few number of stem cells were able to form neurons, and they were able to functionally integrate into the circuitry. Do they protect normal neuron structure also? So these are not stem cells, but these are host brain neurons, what I showed before as a green structure. This mouse was not green, so we stained them with dyes. It's called Golgi-Cox staining. And you can actually see those neurons in the mouse, which is not exposed to radiation here, right? This is mouse which is exposed to radiation, show damage to the neuron structure, which is red. And this is the mouse which is transplanted with stem cells, shows recovery in the neuron structure. So stem cells were able to reverse neuron morphology loss after transplantation. However, stem cell may pose certain limitations because those stem cells don't survive, but we still functional improvements in cognitive function, right? So instead of using stem cells, we started using extracellular vesicles. So what we did here, we took nude rats, or rats we do not have immune system because they may attack stem cell transplantation. instead of taking stem cells, we use extracellular vesicles or EV, or called exosomes. These are nanoscale vesicles which are secreted by several cells. For example, cancer cells, immune cells, stem cells, they all secrete these small vesicles, and they use it as cell-to-cell communication. When they are happy and when they are angry, they actually communicate with each other by these vesicles, and they can carry DNA, RNA, or proteins which can affect the target. So they do not show a lot of immune rejection. There is no risk of tumor formation because this is not cell. And it is very easy to treat those. We can even inject in intravenous injection instead of surgeries to brain, right? So after transplantation of these vesicles, we did cognitive testing to check if they can reverse, if they can show equal effects as stem cell does. We use certain behavioral tasks. Mouse or rats are not as smart as you guys, but you can also check their intellectual ability by performing various tasks, which these are called spontaneous exploration tasks. So what we did simply, we just put those mouse in the boxes with some Lego toys. And we change the arrangement of these Lego toys with respect to type of toys we use, placement of toys we use, and how we expose these mice to the toys. The same mouse and rats can also be tested, their preference to light or dark compartments, their preference to freeze when they have been shocked, or can they swim long or not. You know, there are several tests you can do actually to find out what is their intellectual ability or cognitive function. These are the results. The gray bars here show mouse which are exposed to radiation show decrease in indices of memory. Right? This is memory index, gray bars. Whereas mice which are treated with extracellular vesicles or EV, so significant improvements in those indices. That means which is equivalent to non-treated mice. So not only stem cells, but stem cell-derived exosomes, which are nanoscale vesicles, can also be used to reverse cognitive dysfunction induced by radiation. When we check neuron structure in these mice, as I said here, you can simply qualitatively check here. So this is mouse which is not exposed to radiation or chemotherapy, right? This is mouse which is exposed to radiation. Look at the neuron structure loss. And this is same mouse which is given extracellular vesicle treatment or exosome treatment. You can see reversal of loss of neuronal structure. They gain back their neurons and they gain back their synaptic structure. So it shows the stem cell therapy as well as stem cell derived extracellular vesicles can reverse cognitive dysfunction. When we check neuroinflammation, all the red bars here, which are very high, shows increase in inflammation in the brain. And you can see angry looking microglia here, which is able to attack neurons, could lead to cognitive dysfunction. Whereas black bars are same mice which are transplanted with extracellular vesicles show reductions in inflammation. So not only is preservation of neuron morphology, but also preservation of neuron structure. So what we gain from this entire presentation on stem cells, that stem cells or extracellular vesicles can attain neuroinflammation. It can preserve neurons, which are host. It can also act through various mechanisms that we are currently finding on. So not only you can use stem cells or stem cell or EV, but you can also modulate the radiation therapy itself to make it more safer. So from past 3 to 4 years, in collaboration with University of Switzerland in Lausanne, we started this project called flash radiation therapy. And the basis of this project came from clinical learnings that when you try to treat cancer, there is a fine balance between the type of treatment you choose to. For example, surgery, systemic treatments like chemotherapy, or even radiation therapy. So surgery is the first treatment of choice where when you have defined border around the cancer, you surgically resect that, right? You just take the cancer out and cut it off. However, in certain cases when peripheral border is not defined, you also have to treat that with either radiation or chemotherapy. So nowadays in the clinic, they use combination of radiation and chemotherapy or radiation therapy to treat the cancer. Therefore, fine balance in each therapeutic aspect is important, because not only radiation but also chemotherapy can cause cognitive dysfunction or normal tissue damage. Therefore, advancement in radiation therapy technique is very important in order to preserve normal tissue, but at the same time kill the cancer. So there are two main purposes. You want to kill the cancer, you also want to preserve normal tissue. So those advancements started from many decades. Initially, the only basis was just expose the entire cancer to the radiation. That is called target volume. It was not defined. Radiation therapy was directly used on the cancer that led to a lot of normal tissue dysfunction. Over the couple of more decades, the 2D planning softwares were developed. So you can actually define the contour or border of the cancer to which you will expose to radiation. And further research led to 3D conformal planning of radiation therapy, where you can actually define the 3D volume of cancer with CT and MRI scanning in order to expose those areas of the brain. And nowadays we use more high precision radiation therapy. So recent advancement here led us to think about flash radiation therapy. Flash radiation therapy concept came from the ultra high dosage radiation therapy, where we expose animals to super fast dose of radiation. Now this is the video here. This is the mouse under anesthesia, which has been radiated. You will see small flicker of white spots here. That shows when you use conventional radiation therapy, you can actually see those spots here, right? When radiation comes in. Now this one is flash radiation therapy. Please keep looking at the screen because this is very fast. So flash radiation therapy is super fast, and entire dose can be given in a flicker of a second. So what you saw here was actually a flicker of a second when flash radiation appeared. You can see a flash on your screen. Right? So that's called flash radiation therapy. We utilize this technology not only for brain tumor, but you can also utilize for lung cancer in order to increase when lung is actually breathing. You don't want to miss the cancer, but you also don't want to damage the normal tissue, right? So flash radiation therapy can also be used for such effects. From past several years, University of Switzerland group has shown that flash radiation therapy not only is effective in killing the cancer, but is also protective for the lung, brain, and many other tissues using various model systems they have employed. And from many papers, we have concluded that flash radiation therapy is actually a safer technology. And in our laboratory, what we showed that when you check the cognitive indices, blue bars here, where conventional radiation leads to decline in the behavioral function, orange bar flash does not actually show that. Same dose, same, but less amount of time, it does not show cognitive deficits, which can also be shown in terms of neuron structure. Conventional radiation, loss of neuron structure. Flash radiation, no loss in neuron structure, or no loss in the spine density as well. See the conventional radiation, loss of spine or synaptic units, whereas in flash radiation, no loss of spine or dendritic units. It also reduces neuroinflammation, right? Conventional radiation led to increased neuroinflammation, whereas in case of flash, it did not lead to neuroinflammation. There was no inflammation in flash brain. And this is proposed mechanism where when you irradiate brain, the conventional radiation, which is slow dose rate, produces reactive oxygen species or ROS, which is further increased after a long period of radiation therapy. Whereas in case of flash, there is no production of ROS because it's very transient. It maintains normal oxygen levels in the brain. It kills the cancer, but it does not lead to normal tissue toxicity. And recently, UCI is the first site in the US, which is funded by NIH, to do flash research. Thank you.
So Dr. Daniela Bota has too many hats to actually mention at UCI. Not only is she the director of our Alpha Stem Cell Clinic, she is a neuro-oncologist and clinician here, specializing in glioblastoma. And among her many hats is the organizer and the head of our clinical trial efforts at UCI in School of Medicine, which she's one of our associate deans. So welcome.
Thank you so much for being here tonight. So what I would like to do right now is to tell you a story, and as all the good stories, it has to start with a good title. I'm a clinician. Most of my time I've been a clinician, and every time that I ask a research problem, the problem is inspired by my patients. So for me, while I'm leading an effort to try to prevent or to cure some of the most complicated cancers that people encounter, the glioblastoma, the brain cancer, I have to also think, what's the end result? What's going to be the life of my patient when the treatment is completed? We all ask our family or our friends when they're afflicted by cancer, so how long are you going to live? But as we heard before, maybe the best question to ask is not only how long are we going to live, but how well are we going to live as well. So that's the good, the bad, and the ugly. And the idea that in everything it's balanced, and how can we make it in such a way that we balance quality with quantity and we get to both better quantity and better quality. This is another way to look at the data that you heard before. And this data is updated to what is the projection for us in 20 years from now. In 20 years from now, we will gonna have probably more than 25 million patients that are surviving cancer. And most interesting, 73% of those patients will be older than 65. So when we think about neurotoxicity, now we have to think about neurotoxicity on a patient population that has also a higher incidence of dementia due to the regular life events, due to the fact that memory issues are more common with the older age. So now we have two problems that can compete and compile into each other. So how common is this condition? When we started to study chemo brain, I'm sure you remember, everybody was saying that that doesn't exist. Chemo brain is something that people are just complaining of. They might be anxious, they might be depressed. Of course, it didn't help that the first population that described the condition were breast cancer survivors. And there are a lot of inequities in medicine. Much of the clinical trials, much of the research is done on males. And many times when there are complaints on a female-specific population, instead of being seen as organic and worth treated, we are seeing them as being put on a psychiatric realm. So this is how it started. But in time what we learned is that those young females that were complaining about memory problems were not depressed. They're no more anxious than other patients their age. They're trying to live full lives, but they're trying to live full lives with deficits on memory, attention, information processing speed. So with that in mind, we started to ask a little bit more questions because the number is staggering. Depending on how you measure it, up to 75% of the cancer survivors have some type of memory issues. Just to highlight other things related to the combination between chemotherapy and age, breast cancer chemotherapy increases the long-time risk of dementia by 20%. Studies that were made in aging populations showed that patients that have memory complaints, if you're excluding Alzheimer's and vascular dementia, about 30% of those patients have undiagnosed brain cancer or brain metastasis. And more interesting than that, the cancer itself seemed to cause memory problems. We have modeled it in the lab, and I'm very happy to know that here we have Dr. Lomeli. She has been on my lab for 7 years now. And we are getting animal data confirming the patient's data that cancer in the body induces memory problems and difficulty learning, just by the interaction between the general systemic inflammation and neuroinflammation. So with those data that we are looking here, the next question is, what can we do to change? You already heard about the hippocampus. I just want to remind you that it is a very important structure. We learned about this structure initially because we were seeing this structure developing more and more atrophy in Alzheimer's dementia. Loss of the hippocampal structure directly is reporting to the degree of dementia. Why? We all need adult neurogenesis and we need to form new connections in order to conserve our memories. So I'm going to go back to what I told you before. We go to the clinic, we see patients every day, every week, and sometimes we have amazing achievements. This is almost my only GBM slide for tonight. I'm sorry to disappoint on that. But this is one of my greatest successful stories. This is a patient diagnosed with this very severe brain cancer where people usually live 18 to 24 months. And if you see here, I was still getting MRIs of this patient 7 years later. So I have here a patient that has been in remission for 7 years. But if you look at the red marks, what you see is the contour of the hippocampus. And as you are seeing, as the years are coming and the chemotherapy and the treatments are continued, because this is the type of cancer where you cannot stop treatment, the red structure that was so vibrant here, it's getting smaller and smaller, shriveling and shriveling. And if we take more patients and we average those hippocampal volumes over the years of treatment, what you could see is that if we will put everybody 100% when they started, this is the original volume. By the time that we have completed 42 months of treatment, we have lost more than 40% of the hippocampal volume. I want to kind of let you know that usually on patients with Alzheimer, the loss that you see is 10 to 20% over a decade. And here we are looking at somebody that abruptly lost 40% of the hippocampal volume and was in many cases showing significant cognitive impairments. Yes, my patients were alive for 42 months, but the quality of life was not the one that I wanted to give my patients. So why is this? You heard again and again. The issue is that in the brain, we have the cancer and we have the normal brain tissues. So then the next question that we asked was, in scientific ways, can we make the system simpler? It is complicated. If the cancer is there, you are having surgery, you are having radiation, you are having chemotherapy. Why don't we start by studying patients that don't have brain cancer? They have ovarian cancer or testicular cancer or lung cancer or breast cancer, somebody that their brain is intact when we start the treatment. Can we start by preventing chemotherapy-induced cognitive impairment on that patient population? And with the lessons that we learned from that population, can we then go back to our brain tumor patients, and maybe in combination with improvement on other technologies, we can also address my patient category? So we started with a question very simple. This is a very common drug. We call it Cisplatin. Cisplatin is a platinum-based drug. Very common chemotherapy agents. We are using those chemotherapy agents in lung cancer, ovarian cancer, breast cancer, testicular cancer, in a lot of pediatric cancers. This drug crosses the blood-brain barrier. It does get in the brain, and it accumulates in the brain. And when it gets in the brain, what it does, it affects the DNA of the cells of the brain. How much of it gets into the brain? If you look at this picture here, what you see in red are the area where this drug is accumulating in the brain. And I think that already you have seen so many pictures of the hippocampus that now you're going to recognize this coma-shaped structure. So actually a lot of this drug accumulates into the hippocampus, where it achieves very high and toxic levels. We took then this information and we modeled it in our cell cultures. And what you can see here in this very complicated figure is that cisplatin can affect 3 types of processes. The one that's very important for our discussion tonight is that in high doses, like the ones that accumulate in the hippocampus, it physically kills the neural stem cells. But lower doses will kill part of the neuronal population, and even lower doses will damage the dendritic spines. So what you are seeing here is that a dose that will not kill a cancer cell will be able to kill neural stem cells. So that was giving us a pause, and we started to ask why, what is different. And what we learned that it's different is the fact that the neural stem cells and the neurons are very sensitive to oxidative stress. And the reason for which they are so sensitive to oxidative stress is because the mitochondria on those cells, it's very sensitive to cisplatin and to other chemotherapy drugs. When we are damaging mitochondria, we are getting a lot of free radicals. You heard that very well. And that's when the idea started for us. Okay, so you make free radicals, you are getting memory problems. What can we do to prevent that? Use an antioxidant. We knew about cellular replacement and we knew about all the other things, but we thought what can be simple and easy to move in the clinic. And easy is not easy because it takes years to get from an animal model to people. But the easier answer is we all can buy N-acetylcysteine over the counter. Now, don't go and do that. There are many things that you need to know about over-the-counter supplements, but this is one that it's available. You can go to Walmart and buy it tomorrow. So now we thought about how can we repurpose this dietary supplement in order to maybe fashion it on a treatment that might be able to help us our question. So this is a very strong antioxidant, and it was actually tried in Alzheimer's disease, and it was safely given to the patients, though there were some mild stomach complaints, nothing much, nothing difficult. And most important for us, it was administered to the cancer patients, and it was administered in combination with cisplatin, and it helps some children preserve better hearing because cisplatin also affects the hearing. However, it did not decrease the anti-cancer effects of other drugs. We don't have the luxury of putting chemotherapy on hold while we're giving those drugs. So whatever we give to protect the normal tissue, we have to make sure that it doesn't also protect the cancer. As a side story, I have patients asking me all the time when they take chemotherapy or they go for radiation, should I take high doses of antioxidants to protect my normal tissue? And I'm saying absolutely not. The role of radiation is to create those free radicals to kill the tumor. If you are taking high doses of antioxidants in ways that are not tested and proven in clinical trials, the end result is going to be that you're going to protect the tumor, and you could have a worse response to your treatment and live less. So this is not something that you do at home. But when you do it the right way, what we've seen, and now I know that you guys are all experts on spines, is that if you have a control neuron, has very beautiful spines, the red structures here. If you are giving 2.5 micromolars of NAC, the spines remain the same. My computer didn't contribute very well tonight. With the cisplatin, you could see how we have lost a lot of the spines. And if you're adding N-acetylcysteines, you are having actually a recovery of the spine structures. So, you heard a lot about the fact that our rats like to play. We thought that we're going to give you pictures of the toys. So this is how we train them, and we have great fun in the lab, and there are red boats and green boats. And if you are a good rat, if you have already played with the green boat and you have played a lot with the green boat, and you see the red castle, you'll want to go and play with the red castle. But if you have forgotten that you have seen the green toy before, you're going to keep playing equally with the green and the red toy. So with those very simple systems, we're able to give cisplatin to our group of rats. And as another level of treatment, half of the rats also get N-acetylcysteine after the cisplatin treatment. And what we're able to find out is that N-acetylcysteine is able to correct majority of the learning problems that the rats had when they were treated with cisplatin. So now, what we know is that we have this damage induced by chemotherapy, and this damage can be corrected by N-acetylcysteine without affecting the effectiveness of cancer treatment. The rats that were carrying tumors that were treated with cisplatin and N-acetylcysteine had the same duration of life as the rats that were treated with cisplatin only. So what do we do when we learn about those things in my world? We usually go back to the clinic and say, hey, now is the time. We have the data. We know when and how to give this protective agent. Can we do a clinical trial? Cisplatin is a very, very common drug given to patients with ovarian cancer. What you're going to learn is those ladies, if you keep them alive, they are stage 3 and stage 4 cancer, they start developing memory complaints, and up to 70% of them as well will have memory difficulties. So we designed a study where our patients will receive N-acetylcysteine, and we're going to look at their memory To make sure that we are seeing that their memory is preserved, we'll also look at their MRIs, make sure that we don't see hippocampal atrophy. And we're going to look at survival. And if this study is positive and we are finding that this could be helped with our treatments, then we're going to move to even a bigger study where we're going to have a group of patients that are going to get a control, a placebo, in addition to their chemotherapy, and another group that's going to get our medication in addition to chemotherapy. And you'll ask me, why do you do so many things? Because we really want, when we come to our public and to our patients, to know for sure that what we recommend, it's safe and it's effective. Because this is the most important tenet that we have learned by doing clinical research, that we shouldn't give false promise and false hope, going back to what Dr. Anderson was saying. So, what about kids? Is this different? Is this only a problem of older people? I told you about older adults. Unfortunately, if you have cancer as a child and you get chemotherapy, about 21% of the survivors will have memory problems. And the memory problems will be more significant than the ones that are seen in healthy siblings controls. You know, normal people have cognitive problems as well. They also have anxiety and depression. But what we are seeing more common on our cancer patients that are surviving to adulthood, it's emotional dysregulation. So we wanted to ask the question in the same way, where we treated with chemotherapy infant rats, and we treated with chemotherapy adolescent rats. Now, if you have dealt on your life with adolescents, you will know that they are a very hard group to treat. But we were able to set them down, and we were able to treat them, and we were able to do the research that we wanted to do. So we analyzed all those rats when they became adults. We wanted to be able to compare them with the data that we had from rats treated during their adulthood. And what we discovered is that the young brain is doing very well. The young brain can tolerate chemotherapy from the point of view of learning and of memory. But what we also learned is that the function of the in adulthood is more affected if the rats are treated in the adolescence than the one treated in infancy. And not only that, the rats that are treated in their adolescent time had more emotional problems than either the infant rats or the adult rats, suggesting a more global cognitive dysfunction. Is that because it's the time that the brain finished maturing? We don't know yet, but that's a question that we need to address, and which reminds us that different ages come with different responses to stressors. So, I would like to close here so we have some time for questions. And I want to remind you that it's very complex to understand how the stem cells respond to therapy. And in order to be able to cure chemobrain or radiation-induced cognitive dysfunction, we need to go back to the lab, and we always need to profoundly know what's the biological base for any of those changes. We cannot go directly to the clinic and offer solutions without basic science understanding. We think that we have a way of preventing those changes. Please wish us luck. We are looking at another few more years of clinical trials. And we always think that, as I said before, the animal studies are not necessarily something that it's easy to do. It is very necessary in order to be able to develop the best paradigms for human treatment. So I want to thank you for being here tonight. It has been a long evening, and I'm happy to take any questions.
Thank you so much, both of you. Those are fascinating presentations. I don't know if this is more appropriate for the first presentation, and this may be a very, I don't know, kind of simple-minded kind of question, but how do the stem cells know what to do, right? They go in and they could attack the dendrites or the neuronal hubs or inflammation. How do they know how do they differentiate what they're supposed to do? How does that work?
It's a very good question, actually. So we, when we did our stem cell transplantation studies, we knew which region of brain is most vulnerable. which is most affected in terms of behavior. And we actually transplanted stem cells in those sites where we see maximum amount of stem cell loss. Hippocampus, what Daniela told just now, is highly vascularized. So we have a lot of blood vessels or blood-brain barrier there, which is perfused by blood. And it is more likely to have more reactive oxygen species production because of radiation or chemotherapy. That's why we targeted that region. But there are also other regions of the brain which could be affected by, equally affected by radiation or chemotherapy. So in our first set of studies, we specifically injected stem cells using surgeries into those areas. And they were able to do those things that we expected to do, although they didn't survive. But it also, same systemic therapy affects other regions of the brain. So we came to this exosome concept where you can actually inject intravenous injection that perfuse whole brain. It can go to all the areas of brain and it can equally affect there as well. So it's a technology development over the years. Mm-hmm.
So when you initiated your studies with the N-acetylcysteine, how did you determine the dose to use and how long the therapy would go, when you treated your patients with chemotherapy? What was your starting point?
Excellent question. You always start from the dose that was proven to be safe in other patient population. And there are calculations that you are probably very familiar with where you can extrapolate from a human dose to a mouse or a rat dose. They are available on the FDA website. And then, I don't know, Naomi is laughing in the back, about 5 years of experimentations and many, many, many rats until you get to the exactly right dose. Because it's not only about the dose, it's also about the timing. To give you an example that I haven't included here, if you administer N-acetylcysteine within the first 4 hours, or before the cisplatin treatment, you will negate the effect of cisplatin on the cancer cells. The cancer cells divide very fastly. You need to let them go into a division so that the damage from the DNA gets to declare apoptosis. So if you treat before 4 hours, you're going to basically mitigate the effect on the cancer cells. If you treat after 12 hours, you are not going to be able to rescue the mitochondria from the neurons. So you always have to dissect every of those timelines and make sure that you understand what's the right time to intervene.
Thanks so much for this. It's very exciting work, and I know from the experience of family members how important this area is. I have 2 questions, one for Munjal, one for Daniela. Are the vesicles antioxidants? Is that the way they're working? And for Daniela, I still don't understand why this works. That is, why isn't the tumor protected? Is there some differential uptake of the antioxidant?
So to answer the first question on vesicles or exosomes, it may not be antioxidant according to me because majority of vesicles will, of course, deliver the cargo, but those cargoes should have long-term effects on the system. So what we believe, they might be carrying some signaling proteins, or they might be carrying growth factors and or miRNA. We could change the gene expression of target tissues. And that's how they are modulating the tissue response. So I think it's more associated with molecular cargo compared to the biochemical reaction.
So I will defer this question to Dr. Lomelli, as she spent many, many years answering that question and publishing about it in my lab. So without further ado, do you want to extrapolate on why the cancer cells are not rescued by N-acetylcysteine, but neurons are?
That's a good question. So one of the experiments that we first did to determine whether we could give N-acetylcysteine without reducing the effect of cisplatin on cancer cells was to do a study where we timed the administration of N-acetylcysteine after cisplatin. And what we found was that if we waited at least 10 hours, this is done in with cells in a dish, we could kill the cancer cells without reducing the effect of N-acetylcysteine on stem cells. Now, that was in vitro, so in a dish. So the next experiment was to do this in rats. So for that, we did a study with rats that have ovarian cancer tumors growing. And what we did was after the tumor started growing, we had two groups, one that we were giving rats with cisplatin and another group that received N-acetylcysteine after cisplatin. And what we were doing was measuring the tumors. And we found that if we waited 10 hours, the rats that had the tumors and received N-acetylcysteine were not bigger than the rats that just got cisplatin alone.
And the reason for that, again, what we are trying to explain is that when a cell divides very fast, like a cancer cell, it needs to have the ability of dealing with the free radicals, because otherwise it will undergo apoptosis. So if you don't rescue during the division, the cell will go into apoptosis. Neural stem cells divide very slowly. So by the time that you give the N-acetylcysteine, most of the cancer cells have died, but the neural stem cells are still hanging on, and you can still rescue them before they go into the next division.
Thank you.
It's simple, right?
It's just a few years of work, Come and join us in Sprague. Thank you.
I just so much appreciate your meticulousness and hard work in just progressing healthcare for the folks. Just really appreciate you guys so much. As you guys were sharing, I could see your compassion and heart, and I thank you so much. Yeah.
We thank you.
Any other questions back here? No? These guys don't bite, I can assure you. We have rabies shots close by.
So this is kind of off the wall, but it has to do with radiation. So how are we going to get to Mars, Munjal, and still have a brain when we get there?
Thanks, Brian. So
okay.
there's a reason behind Brian asking this question, because we just don't work on clinical radiation, but we are also funded by NASA, because NASA is interested how space radiation or space travel would affect brain. So as soon as you leave Earth magnetosphere, the entire space is radiation. It's Big Bang, and everything is radiation. So in order to do that, we simulate space radiation. It's called GCR, Galactic Cosmic Radiation Simulation. So it has been done in Brookhaven National Laboratory in New York, Long Island. It's a huge 2-mile big rig where they bombard particles and create space radiation environment. We bring our mouse there. Now, unfortunately, this mice will receive whole body radiation, not just brain. So we have to think about entire body in order to protect that. Now, if you talk about brain, because we work on brain, there are various aspects NASA is interested. It will take about more than 290 days to Mars and back. So it's a long journey. And days, more than that. Registration is open, you can register for that. So during those time of radiation, this small flicker of radiation will be bombarded on the spaceship. And when radiation bombard on the spaceship, hull becomes hotbed. So astronauts inside the spaceship will receive more radiation dose compared to they are doing spacewalk. So we are actually developing more antioxidant compounds. We are also working on stem cell-derived extracellular vesicles, and we are also dealing with some long-term avenues to actually treat this, because not only to and back from Mars, but also when astronauts arrive to the Earth, they should be able to recall and they should be able to function normally. So it actually accelerates the aging in certain cases. So we are long been funded by NASA to develop those compounds. And right now our group has a grant from NASA to just study that. And we have tried at least 2 or 3 different types of compounds. One directly attacks microglia, like Plex compounds. Another set of compound we use NMN drug, which actually affects sirtuin compounds. Similar to NAC affects mitochondrial function, the NMN, which is the precursor of NAD, can actually protect mitochondria and sirtuins and stop reactive oxygen species formation. And third is stem cell-derived extracellular vesicles. There are various things. Problem with NASA is you need to give those pills or whatever drug you want to stay with those astronauts for more than 300 days. So shelf life is good. So we have to think about freezing the stem cells and even lyophilizing them. A drug that should be stable more than 400 days. Can you tell me which drug is more than, no drug is more than 400 days, right? So there are a lot of problems associated with NASA research. So not only finding the drug, but how to save the drug is also important.
And then the drug will be exposed to radiation while you're at it.
That's another thing. So we do 1,000 times radiation on drugs too. Everything, from capacitor, your iPods to switches to spacer, everything is radiated. NASA tests that before they shoot it in space.
When you get back, tell me, and then
I'm not going.
So you pretty much need a replicator. I don't see what the problem is.
It's a good thought, yeah.
All right, well, with that, thank the two of you very much for coming this evening and spending your time here. And thanks to the audience.